Friends, welcome to Biology Made Easy. Today in this video, we'll discuss about cytoskeleton. Now from this name itself, you can guess what we'll discuss. Cyto means cytoplasm and skeleton. So as you know that in case of all vertebrates, there are bones and cartilages to support the body of that vertebrate. In the same way, when the eukaryotic cells start growing, they become bigger and bigger, and there are more membranes formed due to different organelles, it became necessary to give some support, to give some framework to support them. So ultimately, inside this cell, there is formation of certain network of proteinaceous filaments to give support to the fluid matrix, and this collection of this membranous network of proteins is known as cytoskeleton. So if somebody will ask you what is cytoskeleton, a network of fibrous proteinaceous filaments. That means a network of fibrous proteinaceous filaments. Filaments is known as cytoskeleton. Its chief function is to give mechanical support to the cell, and in addition to that, it also helps in locomotion and cell division. Now this cytoskeleton is mainly of three types: microtubules, microfilaments, and intermediate filaments. So usually these are of three types. One is microtubules, and the second one is microfilaments, and the third one is known as intermediate filaments. So we'll discuss one by one. Let us discuss the first one, that is microtubules. The microtubules were first discovered by Robertis and Franchi in 1953, and they first called it as neurotubules. Usually, these microtubules are present in almost all eukaryotic cells. In addition to eukaryotic cells, they are also present in different organelles like cilia and flagella. In addition to the eukaryotic cells, they are also present in cilia and flagella, then also in the centriole and basal bodies, and also mitotic apparatus, then sperm tail, and also they are also present in nerve cells, and they are usually absent in case of prokaryotes. Now if we come to the structure of these microfilaments, usually we will discuss the structure. Usually these are unbranched, hollow, and cylinders, and they are usually 25 nanometer in diameter, while 0.2 to 25 micrometer long, and they are usually made of a protein. Tubulin, And these tubulin monomers, they can assemble and deassemble during growth and dissolution of different substances. That means they can easily, the monomers can easily assemble and disassemble, during quick growth and dissolution. Now, each microtubule consists of 13 protofilaments. And in each protofilament, the tubulin protein is of two types, alpha and beta. Contains alpha tubulin and beta tubulin, and they are present alternately. They are present alternately in a helical manner, just like this. Suppose there are 13 protofilaments. These are 13 protofilaments. Suppose this is alpha tubulin. Then the next one will be beta tubulin. Then this is beta tubulin. Then next will be again alpha tubulin. Again alpha. So likewise, now you see there are 13 protofilaments, and they are present alpha beta, they are present alternately, so that it forms a cylinder-like structure like this. It forms a cylinder-like structure. If you draw like this, it will give rise to a cylinder-like structure. Alpha beta present alternately, and usually actually this alpha tubulin and beta tubulin, they form a alpha beta dimer. It is also known as heterodimer. That will say both one alpha and one beta, they combine together to give rise a dimer, and this is the structure of this tubulin. While alpha beta, they are present alternately in a helical manner and give rise to a cylinder. So this is the structure and where they are present. Now let us discuss the different functions. So among the different functions, now the most important function is it provides mechanical support. That means it provides a rigidity and shape to the cell. Mechanical support. It gives a mechanical support. That means shape and rigidity. The second important point is it helps in locomotion. Because it is present in cilia and flagella. That is why it helps in locomotion. The third point is it helps in cell division. Because it gives rise to the mitotic apparatus. That is why it helps in cell division. It also helps in intracellular transport. Which also we can call as microcirculatory system. It also helps in orientation of the different organelles. There is orientation, or you can tell orientation of cellulose fibrils. In plant cells. And another point is it also helps in cell polarity or determines cell polarity. So these are the important functions, but these first three are very important functions because it helps in locomotion, cell division, and also it provides mechanical support. So this is the microtubule. Now let us discuss the second one, that is microfilaments. So the second one is microfilaments. Microfilaments were discovered by Palevis and co-workers in 1974. Now usually the microfilaments are present in almost all eukaryotic cells, but absent in case of the prokaryotes. So usually they are present in all eukaryotic cells, and they form an extensive network in cytoplasm, and they are more abundant in muscle cells. They are abundant in muscle cells, and that is why sometimes they are also known as myofilaments. They are known as myofilaments because of this name. Now, what is the structure? Like the microtubules, these are also unbranched. But microtubules are hollow, but these are solid. And they are cylindrical, but these are rod-like. These are rod-like fibrils. And their diameter was around 25 nanometer, but here the diameter is around 6 to 10 nm diameter. And the length, in that case the length varies from 0.2 to 25 nanometer, but here it is indefinite length. So mark the difference. Both are unbranched, but that is hollow, this is solid. That is cylindrical, this is rod-like. That is around 25 nanometer in diameter, but this is 6 to 10. And that is again 0.2 to 25 micrometer length, but this is indefinite length. And the microtubules are made of protein tubulin, while these are made of protein actin. These are made up of protein actin. That is why sometimes these are also known as actin filaments. So these actin filaments, they may be present either singly or they may be present parallel in bundles. So this is only the structure of this microfilaments. Now let us discuss the functions. So the microfilaments have a lot of functions. And number one function is cytoplasmic streaming. As you know that all eukaryotic cells, the cytoplasm is not constant, it is always in a state of motion. So that is known as cytoplasmic streaming. And this cytoplasmic streaming is due to presence of these microfilaments. It is a very important phenomenon. That is why the cytoplasm always remains active. This is the first point. Then the second one is support. That again support to the matrix of the cell. like that of the tubules. Then the third point is it helps in formation of pseudopodia. Formation of pseudopodia. So usually those who move through pseudopodia, like amoeba, that is always sol-gel interconversion. And the sol-gel interconversion is due to this microfilaments. That means this is due to sol-gel interconversion, it is possible. Then the next important point is again in case of leukocytes and macrophages. In case of leukocytes and macrophages, there is also, you will find that is amoeboidal movement. Amoeboidal movement. So that is also due to the presence of these microfilaments. Then the next point is that is muscle contraction. Muscle contraction. Then the cell membrane movement. What is the meaning of this? That means during endocytosis and exocytosis. Endocytosis and exocytosis. So usually what happens? When there is suppose this one cell membrane, and suppose one solid particle will approach it and it will enter inside. So naturally here one vesicle-like structure, that is the phagosome, is formed. So this is possible only due to this microfilaments. So that is also one of the important functions. Then another function is furrow formation. Furrow formation during animal cell cytokinesis. Animal cell cytokinesis. Again, when there is in case of animal cells, when there is cell division, the nucleus divides in two. Then the cell membrane constricts during cytokinesis. So this constriction is due to only these microfilaments. So that is also one of the very important functions. Then the another function is that is the movement of cell components. Movement of cell components. That means inside the cell different organelles are there, different pigments are there. So it is the duty of these microfilaments to keep them separate from one another, so that there will not be any interference between one another. They will keep them separate. So these are the important functions of the microfilaments. Let us discuss the third one, that is intermediate filaments. So the intermediate filaments, their size is almost intermediate between these two, that is microtubules and microfilaments. And instead of a single protein, they consist of different types of proteins. The third type is intermediate filaments. Intermediate filaments. So usually it was discovered in or first reported in 1968, and these are usually present in all eukaryotic cells, but they are mostly absent in plant cells. Absent in plant cells. Plant cells. Usually, like the microtubules and microfilaments, these are also unbranched. These are solid like that of the microfilaments, and these are usually non-motile proteins. So what are the different types of proteins that are present? The proteins are usually the proteins which is present in intermediate filaments proteins like that is keratin, desmin, and also vimentin. These are the different types of proteins they are present. And depending upon this, if we come to the structure, the intermediate consists of 8 protofilaments. 8 protofilaments. And if you draw a structure, it looks like this. These are 8 protofilaments, and it will give a structure like this, and which is like this, a rod-like structure, and it may be made of protein either keratin or desmin or vimentin. Now, this intermediate filament is again of 5 different types, depending upon usually what type of protein it is. So there are types, and usually there are 5 types. 5 types. So number 1 is usually keratin. Keratin containing tonofibrils. Tonofibrils of desmosomes. Desmosomes and keratin of skin. That is number 1 type. The number 2 type is desmin. Then desmin containing muscle cells. Desmin containing muscle cells. Then the third one is vimentin containing. Vimentin containing, these are usually widespread in case of that is a nuclear membrane. Nuclear membrane connects to cell membrane. Then the fourth type is neurofibrils. Neurofibrils. So these are usually present in case of nerve cells. And the fifth one is glial filaments. Glial filaments. Usually these are found in astrocytes. Astrocytes. So this is the structure, and these are the different types. Now if we come to the function, what are the different functions carried out by these intermediate filaments? So we'll discuss the functions here. So one of the function is support to nuclear matrix. Then again the second one is again support to the fluid cytosol. The third one, it also gives support to the epidermis. So it is support to the epithelia. Support epithelia by binding to spot desmosomes. Then another function is it provides strength to axons, and naturally therefore due to the nervous system or nervous tissue. Then the keratin supports skin, and the cardiac muscles. The cardiac muscles are interconnected by spot desmosomes, which are made up of intermediate filaments. So these are the three types of cytoskeleton: microtubules, microfilaments, and intermediate filaments. And in each case, where they are present, what is the structure, what is the difference between them, and what are the different types of functions. The cytoskeleton is very important from examination point of view. So thank you very much for watching the video. Wish you a very bright future. God bless you.